So a very good morning to you all and welcome to our biology class of today. I'm Teacher Pacific. Today we are going to correct together this Cambridge IGCSE Biology Paper that was done May June 2024 with code 0610/61. It's a paper 6 which is alternative to practical, and we did it here at BA as our paper 6 exam. So I want you to have your paper with you and we do correction in a very short time. Now, the first question, cooking fruits and vegetables in water reduces the vitamin C content. The vitamin C is transferred from the fruits and vegetables to the water by diffusion. We pay attention with this one because you understand the content of vitamin C will be moving out of fruits to our water content by diffusion. Now, DCPIP can be used to estimate the concentration of vitamin C. The blue DCPIP solution reacts with the vitamin C solution and becomes colorless. This is exactly how we test the presence of vitamin C in something. We always use this reagent, which is DCPIP, which is blue in color. And if vitamin C is present, we know it is going to turn into colorless. Always, like we said, we should pay attention with this very first sentence. A student investigated the effects of cooking time. The moment you see the word effect, the next part of it will be our independent variable because it is exactly what we are changing. If we say the effects of cooking time, I see if I use a short time, what is going to be the vitamin C content? If I use a longer time, what's going to be the content of vitamin C? So we'll be changing the time, then we measure the other part that we talked about. And now you can take enough time and read, like you have a paper in front of you. And this is the results that they got at the end. They saw that the volume of DCPIP remaining in the gas cylinder, and remember we are changing time. I think we are seeing this is our independent variable. We are seeing if you use 1 minute, what is the volume of DCPIP remaining in the cylinder? You see this, the one. Means we add this one for it to go back to 5 because we always refill to go back to 5. All right? If you use 2 minutes, you can see that it is using, we have now 2.6, we are adding in 2.2. This is exactly what is remaining. That means the more time we use, then the remaining volume of DCPIP reduces. All right? You can see it. Now, using the equation, calculate the volume of DCPIP added to the sample taken at 5 minutes. Now, at 5 minutes, we can see it is here. Now, the volume of DCPIP added is the starting volume of the DCPIP in the cylinder. The starting volume is so that is always 5. And the moment you use it, we always refill and we go back to 5, okay? Now, you can see the starting one is going to be 5. Then we can check on the remaining one is this one because the formula is telling us that starting volume of DCPIP in the cylinder minus the volume of the DCPIP remaining means it is minus 1 pointer. It is minus 1.6. Then you can work it out and you will see that it is something around 3.4. I saw that many of you can easily do it. And it's exactly this that I'm talking about. The starting one, which is 5, and the remaining one, then you have 3.4. You can do correction on it. Now, prepare a table and record the result shown in figure 1.1 and your answer to this. Now, we see the table can be like this. Always, I said it, the independent variable is always here. This is exactly what you're changing, which is time. And also here we have the dependent variable. The rating is not perfect, but I hope it's clear to you. So we can see it. We are just filling in the numbers that we have here. But also there is this answer here is really perfect. It is really perfect. So what we did, we have time, which is an independent variable, and also we have the volume of DCPIP, which is being added. This one is also 100% correct. Now, state the conclusion. Now, what is the conclusion in regard of what you have here? The conclusion is, as you see, the longer cooking time, because we can see that here the time is increasing as we're changing it, allows more vitamin C to diffuse out. And the reason why we know that vitamin C is increasing and diffusing out, we can see that the volume of DCPAP added is also increasing. So that one, I think it is clear to everyone. All right. Now, state the independent variable in this investigation. Independent variable, like we said, we saw that it's exactly what you're changing. You can see that we're talking about the student investigated the effects of cooking time. Means our independent variable here is going to be time. Time. Because it's exactly what we are changing. I think we saw it. Now, state the purpose of wrapping the beaker in the foil in step 1. Now, like we know, the reason for that is simply to decrease heat loss. We don't want to decrease heat loss. And also, maybe you can even have other answers. We can say for insulation or to maintain temperature. Because here we saw that it is just time. And also here, talking about this one right here, we're going to see that it is to decrease heat loss. We don't want our heat to be lost. And also it's for insulation. I hope that one also make it right. If you didn't make it correct, then you can do correction. And also explain why it was important to remove the air bubble from the cylinder. Always, before we use the cylinder like this one, like you see here, I'm measuring the volume. But we should remove all the bubbles and everything in the cylinder to make sure that the volume of DCPIP is accurate. It's very important. Air would affect our volume. So we have to make sure that before we use a cylinder, we remove all the air outside. So the volume of what we measure is accurate. So I hope that one also is clear so that many of you did it right. I hope you will all just pass and do correction. Now, the question I want to take on more of my time is this one because this is a random question which is always there. And we should always get 6 out of 6 in this because it is very easy. It is a repeated question. And it is one of the questions that you come in our paper knowing that this one is coming. So, plan an investigation to determine the effects of light intensity on photosynthesis in an aquatic plant. Like we know, if we are planning an investigation, we are always starting with an independent variable. What are we going to change here? Like I said, if you read the word effect, you know the next thing to it. Means here we are changing the light intensity. And then we measure the volume of oxygen produced, so we see how photosynthesis, the rate of it is happening. Now, for the independent variable, or what you are changing, it is going to be different light intensity. We are going to see it. All right? We are changing the light intensity. Dependent variable, which is our DV, is going to be what you measure on photosynthesis all the time. On photosynthesis, we measure the volume of gas given out. Because there is always, when photosynthesis is happening, there is oxygen gas which is released out. We measure the volume of it. That means the volume of this oxygen is our independent variable. We are going to sit down because I have where I prepared, and maybe we take some notes. But I want you to exactly understand what I'm talking about here. Now, the third point, we are going to get it from rating the method. Now, talking about the method, for photosynthesis, it is the same thing. Nothing changes in it should. The first thing, the method of changing light intensity. So I saw many of you could get 4, 5 out of 6, 3. Make sure that you rate your method very well. Means the first thing you talk about is changing the light intensity. How do we change the light intensity? We saw that we changed the distance of the lamp from our plant. And this is in centimeters because I saw where some people rate 30 meters, 40 meters. That is really not right. Now imagine a light in 30 meters in the lab. I think it can't make no sense there, right? So, but if you say we are going to put our lamp from 10 centimeters, 20 centimeters, 30 centimeters, you are changing the light intensity there by different distances from the lamp. Talk about correcting gas in a gas cylinder. That is also a method you can use. Means you say changing the light intensity by changing the distance of the lamp from the plant, all right? And you measure the gas given out in a gas cylinder. The third thing, always when you are investigating photosynthesis, there's something that you need to pay attention with. The first one comes with avoiding heating effects of light. I talked about it. So I want us to check together on this different PowerPoint that we have here. Exactly this one. Exactly this one. Here I was investigating the effects of changing light intensity. If you check very well, let me put in full screen. Yes, if you check very well, we can see what I can be doing. I have a ruler. So this one is for me to change the distance of the lamp to my plant here. All right? Means I may say in 10 centimeters, in 20 centimeters, in 30 centimeters. I make sure that I move it, yeah? But there is something important. These lamps right here, like this one here, can release heat. As much as we need light, it can also release heat. And we need to have the same heat. So this is why we put in this glass tank filled with water, so that water absorbs the heat and only light passes. The way we solve this usually, we don't use lamps in the laboratory. We use what you call LED light. My handwriting here. LED light. So for the LED light, only releases light. So means we are changing only light intensity. All right. By this one, if you use a lamp, you should put it in your method how you are going to avoid this heat reaching here. So we can put here a glass of time. Means this one is going to absorb all the heat and hinder the heat from moving from here, going all the way to the plant. But heat will stop here, and only light, which is the only changing factor we have, proceed going there. So we can improve that by using LED bulb or light lamps, and those ones help to stop heating from reaching there. We have other points of we can investigate, but today, since it is light, let us only focus on light. All right. So we've talked about all those points. There is a independent variable, dependent, the method. We saw that we can method the changing light intensity, method of collecting gas by using a gas cylinder, and also a method of avoiding heating effects of light. Means we can use heating shield or we use LED light. And also after that, we also go to where we get the fourth mark from. Means we talked about IV, which is the independent variable. Dependent variable. I talked about the method, all right? Now the next one should be your control variables. Control variables should always be 3, like we said. Since I'm changing the light intensity, I should exactly know what I'm going to control and keep the same. It can be the temperature, because we have 3 most important things that can act as limiting factor for photosynthesis. That is temperature, light intensity, and carbon dioxide. That means for the control variable, I can keep temperature the same, I can keep the same species, I can keep the same concentration of hydrogen carbonate. Why the same concentration of hydrogen carbonate? It's because hydrogen carbonate give out carbon dioxide. Yeah, that one also we should know it, and I can keep the time the same. After my control variables, I hope in your correction I'm going to see all these things on your paper. Make sure you write temperature, write time, write hydrogen carbonate, because it is the one which is the source of carbon dioxide. So the next thing that we talk about here is repeating. Repeating, you don't say I'm going to repeat my investigation. No. I will repeat twice or thrice. You should repeat like 3 times and calculate the average of the volume. All right? And lastly, you talk about safety. Safety precautions. And since we are dealing with plants and all that, we can use gloves and all that. So, all the patient don't change anything. Talk about your independent variable, which is what you're changing, dependent variable, which is what you're measuring, method that we're using, and also we use the control variable. They should always be 3, and always you know what you can keep the same when you read the question. But for photosynthesis, we have 3 limiting factors. You should follow the video on photosynthesis. It is light intensity, if it is what I'm changing, so temperature should keep the same. Hydrogen carbonate, which gives out carbon dioxide, should keep the same. Time and all that. And here is some sample of what you can write. Like here, the method. Maybe we go back and use the one we used. but it can also be using different lamps. Distance, yes. Correct gas using a syringe. The plant is supposed to be submerged in water. That's very important. And also, we saw that you can use LED light to avoid that lamp releasing heat. Control variables, there should always be three, same type of plant, same time, same temperature, same amount of hydrogenocarbonates. We repeat 3 times, but here you should add on and calculate the average, and at the end, you use your gloves. There is another one we can use here, and you can see here it is independent. We are changing the distance, which is an independent variable. Dependent, we are measuring the volume of a gas in a gas cylinder. Very important to say what you're going to use to measure. We don't say we are going to measure gas only. You measure gas with what? All right? And also use sodium hydrogenocarbonate. This one is the source of carbon dioxide. So you understand that in photosynthesis, carbon dioxide is raw material. So it should be there. Using the same temperature, it's very important. Repeating 3 times and look for the dip. And also safety, use gloves. It's exactly this way that we test carbon dioxide. Here we are measuring, right? We are measuring. And you see, the gas cylinder, we saw that we should first remove all the air so that our volume don't be affected and all that. This is another question which is always there. We only pay attention on details. We make sure that it is like this. From what you saw, lines which are like this, they are not allowed. So if it is a line, let it be like this. It should just be a one-way line. And also, we don't shade anything, all right? And we pay attention on details. Like here, you see that we have 6 seeds, but we have 2 where we have no seeds. And also, these very details, they should be included in your drawing, like this one exactly. I repeat again, don't shade. And this one should take at least 65% of the space given. So you don't draw a small one or which is not clear. It should be clear, but you pay attention on details, and it should be one-way line, mostly on the surrounding of it. Don't shade anything. Don't shade anything. This one we are going to measure. You just use your ruler. I think this one also is one of Venne's equation. You're going to use your ruler. You measure P to Q from here to here, and after you can see that maybe on marking schemes we have something that might be different to what you have after printing out. There are those ones who got 95 because of how the paper is printed out. So you understand here you should be able to calculate the actual distance, because it is what they're asking. What's the actual distance? Like we said, there is something we call I am. Now, since you are looking for actual, then actual is supposed to be equal to image over magnification. That will be 95. That will divide by 2.2. 2.2 is our magnification here on the equation. I think you can see it here. It is right here. Over 2. It's 2.2. All right. So the answer is supposed to be 43 point a lot of things. But always pay attention on these details. If they said it should be 3 significant figures, then let it be 3 significant figures, right? So it should be 43.2. But on the marking scheme, you might find a different answer depending on how it is printed. Now, plants can make fats, starch, and proteins. These are biological molecules. State the names of reagents used for starch. We know this one is either. This one is iodine solution. It is the one we use. And you know it moves from brown to being blue-black. We can use also proteins. If we talk about proteins, then the burette, we use burette reagent. This is the reagent that we're going to be using. And it is going to also show us the color change. And for that one, we know the color and how it changes on all that. It will be a class of another. Now, describe the method you would use to test the sample of plant tissue for fats. How do we test fats? This one is called immersion test. We can see it here. There we used iodine solution, and also we have burette solution. These are reagents we use to test them. But if they ask us to describe the method you would use to test a sample of plant tissue for fats, the first thing, you add ethanol to the solution or to the sample that we have. Then we put it in water. Water that we use, then we shake. It's very important to mention that. It is not in our answer that we have here, but it's very important to shake. And also, White color appearance, and this is what you call immersion, shows up if you have positive test for it. So we should make sure that we be careful on it as well. All right. Now for C, it's another important question. The enzyme pectinase is used to produce apple juice. Pectinase breaks down the cell wall of apple tissue. This increases the volume of juice that can be extracted from the apples. Now we are here. Very important, like we saw the word effect. I told you what is always next to the word effect. A student investigated the effects of the concentration of pectinase. That means this is our IV. It is the independent variable, and we are going to change the concentration of pectinase on the volume of juice extracted. This is going to be our dV, which is our dependent variable. All right. So we are proceeding on where we were. Uh-huh. So on this one, we saw that the enzyme of pectinase is used to produce apple juice. For this one is a very important question that also we should understand very well, very well. And also we are from seeing that this one right here is exactly what I'm changing, and this is exactly what we'll be measuring. I think it is clear. Now, the mass of 7 samples of chopped apples were measured. The sample was then put into a different beaker. Means we had seven samples. Now 50 centimeter cube of pectinase, this is a volume of pectinase, you see it is the same in 6. That means this one is what we are keeping the same. It is a control variable. It's a control variable. I want to think that it is clear to us that it's a control variable. Now the beaker were kept in a thermostatically controlled water bath set at 37 degrees Celsius for 30 minutes. The content of each beaker were filtered through the filter paper, and the liquid was corrected in a measuring cylinder. The volume of liquid was each measuring cylinder was recorded. Now state the dependent variable. I told you this is exactly what we are changing. We are changing the concentration. We should always read the moment. In this sentence always gives you nearly everything. This is our independent variable because it is what we are changing. And also we have the volume of juice, which is the dependent variable, which is what we are measuring. I want to think that it is clear to us. Now, that means for dependent variable, it is going to be volume of juice filtered in the liquid. State the 2 variables that were kept constant. There we have very many. We have the volume of pectinase. It is the first thing. We are using the same temperature in all the 7, because if you don't use the same temperature, then it won't be a fair test. All right. So we are using the same temperature. All right. We are using the same volume of pectinase, same type of fruits. It's very important as well. Same incubation time. We should use the same time as well. Now, this one we talked about it. I hope now you can easily describe this method by adding ethanol to the sample. Then you put in water. Then you shake. Then what you're going to see it is the white color appearance, which is the immersion test as well. The immersion appearance as well. Now, for the hazard, identify a hazard in the method described in 3C and suggest a safety precaution to reduce the hazard. The hazard is enzyme, because we are dealing with enzymes here, and also the precaution is that you can always use gloves. So as you do correction, I hope some of you did it right, so you can still do correction here. A student did not repeat the investigation and only collected one set of results. Explain why it's better to collect several sets of results. Here is to identify anomalous results, to see if there is a result which is not fitting the pattern. Okay? I'm moving fast. The seventh beaker of chopped apple. Remember, we put in 6. We didn't put in pectin solution. State what should have been added to this beaker to make it a control experiment. If we are putting, always what is the role of the control experiment? It is to see if we don't have the independent variable, how the experiment can go. Means in all the 6, we did put in pectinase, but of different concentration. Now for a control experiment, we are not putting in any enzyme. We can put in water. All right? We can put in water. We can add in water. We can also put in denatured pectinase. Why denatured pectinase also cannot work? Anytime they talk about the control experiment, remove the independent variable. I repeat, anytime they ask you a control experiment, remove what we are changing. Means if we know for the reaction to happen, we should be having the enzymes, which is pectinase. Maybe what you're changing is the concentration. For us, you're going to remove it. We can use water, we can use denatured pectinase because they are enzymes and all that. Another easy question. A sample of 150 grams of chopped apples was placed in a beaker with 0.8% pectinase solution. We can see it here. Okay? But now here, it is in 1 gram. People, I want to always, whenever they ask you these questions of calculating, to remember that it's in 1 gram. Now someone is asking you to put in 150 grams. It's as easy as that. A sample of 150 grams was chopped. Of chopped apples was placed in a beaker with the 0.8% pectinase solution. Using the information in table 3.1, calculate the volume of liquid corrected in the measuring cylinder. Remember, here for us we are correcting per 1 gram. For them they are telling us 150 grams. So what we do is simple. Means we are going to take 150 grams, we multiply by 0.92. It's as simple as that. And the answer will be 138 centimeter cube. This is it. This is it. Always when they ask you, they were changing the grams. For us, we are dealing with this 1 gram in 150, then you had to multiply it since it is in 0.8%. On the graph, it's very important for 4 marks. Remember, we just need to copy this. What you have here, we come and put it here. We have our x, or raise, it's what we have to start with. You put your line here, x and y. All right, we have a volume of liquid corrected. Volume of liquid corrected means this one, it is an independent variable, like we said. It is always on x-axis. Never forget to write this because we get some marks here. And also you take the second column, which is dependent, you write it here. Make sure you have good scaling. We have 0, we don't go to 0.5. It is 0.0, then you have 0.2, 0.4. You see that they increase. We have the same jump that we have. Intervals are the same. And also even on x, intervals are the same. So it means we are not changing anything. Then at the end, we are going to make our graph, which is straight. And also they're asking us to describe the relationship between the volume of liquid corrected from the chopped potato apples and the concentration of pectinase. This one, from what we see, is that as the concentration, which is what we have here, as the concentration of pectinase solution increases, because you can see that it is increasing, the volume of liquid corrected increases too. Means here it's from here up to here. Then levels off at 0.8% of pectinase. This one, never forget to mention that it levels off. Never forget that. So that is, guys, make sure that you do correction of your paper, complete it very well, and the most important thing is understanding each and everything. So thank you so much for following. So see you in the next one. Bye.